My name is Jocelyn Richardson. I'm an associate scientist here at SLAC. My research is primarily in understanding how plants uptake nutrients and heavy metals from soil so that we can think about ways that we can sustainably increase nutrient uptake by plants without the need for things like fertilizer. We all know that plants need certain nutrients just like we do to survive. Plants get those nutrients from the soil, where their roots are basically facilitating this uptake of these nutrients. Nutrients are present in soil when we think about things like phosphorus, sulfur, potassium, and then even metals like iron and manganese. However, some of those nutrients themselves are present as soil minerals, and that is considered not available to plants or even microbes. And so various different types of processes have to happen within the soil in order to make that nutrient bioavailable to the plant. And so I studied those specific processes from going from something that's considered not bioavailable and how it becomes bioavailable to the plant. So we can think about how we're able to improve on these processes ourselves by our own intervention to help those plants uptake those nutrients, to make sure the plants are growing, they are prolific, and especially thinking about helping them be able to grow in the face of climate change, thinking about increased temperature, drought, or even severe precipitation events. So here at SSRL, we have a facilities grant by the Department of Energy's Biological and Environmental Research Division. Since I started about 5 years ago, it's been my role to be able to facilitate increasing this type of research into plants, beneficial soil microbes, and soil with a variety of different collaborators. And through that effort has also borne my own research area, particularly focusing on potassium uptake by plants. Well, in particular, I really use the synchrotron a lot for my research. The particular X-ray techniques that we have here facilitate being able to visualize in situ the nutrients that I care about. And so I don't have to necessarily alter the samples in any particular way. I can directly image particular nutrients within different parts of the plant, so plant leaves, stem, even in intact soil cores. So I can really look at the distribution of these nutrients as they are in the environment right here in the lab. In the future, we'll be procuring a plant growth chamber so that we can actively grow these plants at SSRL, and that will enable dynamic studies of these nutrients. So growing the plant in a growth chamber, bringing it to a beamline, measuring the nutrients in situ, and actually returning it for plant growth in the chamber, and repeating those experiments so that we have a better understanding of these long-term nutrient uptake by plants, which is something that isn't actually very common at any synchrotron facility. Honestly, I really enjoy learning. I think every day that I come to the lab, I am learning new things. Even though that this is my particular area of research, every data set that we get on a different plant or a different microbe is learning something new. And I think we need to be able to harness that knowledge in various different types of ways in order to really pursue a more sustainable understanding and effort for everyone worldwide.